Okay, so what we're scheduled to talk about today are those two papers that I posted from the Gary Ackers lab. So this 1991, excuse me, PNAS paper and this 1992 science paper, okay? And so I just put this PowerPoint out there. I was working on it through yesterday. I'm still not happy with it, but there it is. So as usual, this is a little bit in the way. Okay. So the Ackers lab from the early 70s, I guess, was working on hemoglobin and developing methods for analyzing statistical thermodynamic data, okay, along the lines of what we just derived from Minot, Wyman, and Changeau. That was the kind of thing that they were developing. And they were basically trying to understand oxygen binding to hemoglobin. It's not the only system they were working on, but that's what we're going to talk about. And these two reviews were basically 20-year summaries of where they were at that time. Okay, now I want to go back to a couple of slides from the hemoglobin lecture when in medical school, okay, in the medical class in biochemistry. And this is one of the slides that I showed. I've modified the text a little bit. So this is a rendition of the myoglobin structure. It mentions that it was the first protein structure, high resolution structure, crystallized. The first author on that 1958 paper was John Kendrew. And I mentioned that it was a paradigm for alpha helices and hydrophobic residues being on the inside and polar charge residues on the outside, etc. What I've added is this last sentence. This last sentence, I don't think I mentioned it here before, but I mentioned that both John Kendrew and Max Perutz got the Nobel Prize in 1962 for their studies on the structures of hemoglobin and myoglobin. So they were both at the same place in Cambridge in England. A new facility for crystallography that was set up. This was basically the same place where Watson and Crick were working. And they, Kendrew crystallized myoglobin, and a few years later, Perutz successfully crystallized hemoglobin, and then his lab crystallized both oxy and deoxyhemoglobin, and that sort of is the beginning of the whole field. Okay. Here. This is taken off of a wiki page for Perutz. His name is Max Perutz. If you go looking for this, at the top here is what I just said, basically. Down below, it describes some of the things that he's worked on. And it mentions at the top structure of oxy-deoxyhemoglobin at high resolution. So in bold, as a result, in 1970, he was at last able to suggest how it works as a molecular machine, how it switches between its deoxy and oxy states, in turn triggering the uptake of oxygen, and then it's released to the muscles and other organs. So the lecture that I give you in medical biochemistry is concerned with integrating protein structure, hemoglobin structure, with transport of oxygen from the lungs to the tissues, and the reverse transport of CO2 from the tissues to the lungs. And it's sort of focused on getting you to a point where you can understand arterial blood gases and interpreting arterial blood gases and the role of hemoglobin in that process. Okay. So it's this last part of the sentence. All right, triggers uptake and release of oxygen. But in 1970, Perutz basically was looking at crystal structures. And he was saying, here's the oxyform, here's the deoxyform. Let's look at the differences and explain how this machine, molecular machine, works. And he basically described in some molecular way what had to move where to make this happen. And that was sort of the level at which he was making this description. I'm making it a little bit shorter than it really is. It's much more complicated than this, but it's not really a full-blown statistical thermodynamics model. It's not really an understanding of the principles by which, the code by which oxygen binds and hemoglobin binds and releases oxygen. So around this time, or in the 90s, basically, when Gary gave a lecture, and I've saw him give this lecture a couple of times probably, or lectures like this a couple of times, he would use this example. These are cartoons from the Shakespeare play Hamlet. And the opening scenes in Hamlet are a wedding. Hamlet has come back from wherever he was, the Prince of Denmark. His mother has married someone else. His father has died. He doesn't know under what circumstances his father has died. And he's not happy with the fact that his mother has married. There's a new king, consequently. But the opening scene is basically a wedding. The closing scene of Hamlet is a lot of death. Hamlet dies, he kills his mother, he kills the king, his sister has drowned herself, etc. Okay? So think of this as deoxy and oxyhemoglobin. If I know the structure of oxyhemoglobin and I know the structure of deoxyhemoglobin, I know the story. I understand what has happened. Okay, this was basically where Perutz was coming from. We have the structures, we understand. And what Gary said, and what his entire scope of research was saying, and by the way, it's something like 40 papers at least that contributed to this story. What about the intermediates? What actually happens when oxygen starts to bind? What triggers it? How much energy is involved? What are the processes that go on in the intermediate states? Okay, is that important? Is that part of the story? Are you missing something by not knowing the intermediate states? Okay, I and a friend of mine played with this a little bit. And we do the same thing with Casablanca, the movie Casablanca. In the beginning of Casablanca, there's this young couple in Paris. They're falling in love. They're partying. They're listening to music. They're drinking, driving around Paris. At the end, he's on the tarmac watching a plane take off flying to Lisbon, and he's there with the French inspector. He has just killed a German officer. She has just gone off with her husband, who she thought was dead. Okay, and here we are again. Here's the beginning, here's the end. We know the story. Part of this comes from this concept of path independence. Okay, it doesn't matter how you go from the beginning to the end or the end to the beginning. The thermodynamics of this process is the same. Okay, the overall path is, the overall thermodynamics, the overall energy changes are independent of the path. Okay, so it's the same thing, obviously. There's something in the middle that's missing. It's probably important, but who cares? We've got the crystal structure at the beginning, the crystal structure at the end. We understand the mechanism. Okay, and that was basically Perutz's point of view. And quite bluntly, there are a lot of people today, this is 50 years later, who still think that, who still think that Perutz sort of solved the problem. Okay, Around 1965, this is the paper that we're talking about. Monod, Wyman, and Changeau published this paper on allosteric transitions. And they basically made a proposal. And the proposal doesn't just incorporate hemoglobin. It basically is this proposal for every allosteric protein system. Mostly they were enzymes. Hemoglobin is an honorary enzyme in this scheme. And they created language and understandings and a model to explain this. So they introduced the concept of homotropic effects. The ligand affects its own binding. Heterotrophic effects, other small molecules, other different ligands can also have an effect, positive or negative, on allosteric interactions. The allosteric proteins are polymers comprised of subunits, so there are oligomers. There involves quaternary structural changes. The heterotrophic effects can be positive or negative. They make the assumption that heterotrophic effects are known to be positive or negative, but homotrophic effects appear to only be positive, and so they only incorporate positive effects in this model. There are, in fact, a few systems that have negative effects. And then mutations will disrupt or interrupt or affect the nature of the homotrophic heterotrophic effect, and so studying mutations can be important. Okay, so this is copy pasted out of the 1965 Journal of Molecular Biology paper. They also have these summaries. A polymer protein contains a finite number of identical subunits. The identical subunits are associated with an oligomer designated as protomers. The term monomer describes fully dissociated protomers of identical subunits. We accept the fact that hemoglobin has non-identical subunits, and so we overlook that for the moment. The term subunit is purposely undefined, and it may refer to various submolecular entities. Okay, but basically they're introducing language. And what I want to point out to you is you're familiar with all of this language, but they were basically creating sort of a definition, the language that we should be using for this, you know, some 55, 60 years ago. And then they also had this list of summaries. Allosteric proteins are oligomers. There's an axis of symmetry. The axis of symmetry and the structures were a very important part of this early on, because most of these molecules tend to have some defined symmetry that is maintained. There's some stereospecific complex made with between the ligand and the protein. And something about how those interact specifically with this symmetry. The conformation of each protein is constrained. I think that language constrained is used a few times in the Ackers lecture, but these guys actually introduced this idea of constraining the structure within the protein. And this constraining of the structure ends up being discussed in terms of some kind of cooperative interaction. It can be a positive or a negative cooperative interaction that helps maintain the symmetry. There are at least two states reversibly accessible to the enzyme. This is the introduction to the idea of a T state and an R state. Okay. The affinity of at least one of these states changes for the ligand. Okay, and so in some of the modeling, maybe the ligand doesn't bind to the T state at all. Maybe it only binds to the R state. We know that that's not true, but it's high and low affinity. Maybe the hetero specific regulators only bind to the T state, not to the R state, et cetera. Those were possibilities that are discussed. And when the protein goes from one state to another, its molecular symmetry is conserved. Okay, and so all of these ideas, if you think about these and read these are sort of general ideas that basically was proposing a model for fitting this. Now, I didn't copy paste it. There's a one-page derivation of what we went through to derive the Minot-Wyman-Changeau model. And they basically derived that model. A lot of the details are sort of left out. They start out with the idea of ligands bound per total proteins, accept the fact that there are two states, a T state and an R state, and then go on to define the equation. There's an L for equilibria between the two states. There's a C for different affinities, etc. Okay. And so what we have in this paper, and I'm actually starting with the 1992 science paper here, is models for cooperative binding, but specifically the molecular code in cooperativity and hemoglobin. And what Gary's work did, and what the work of all of these people in Gary's lab did, were define this in far more detail and dig down into the details of this, okay? And so, is that clear? And so this is just zooming in on the abstract. Recent experiments on the energetics of cooperativity, along with assignment of their quaternary structures, have revealed that the allosteric mechanism is controlled by a previously unrecognized symmetry feature. Quaternary switching from form T to form R occurs whenever heme site binding creates a tetramer with at least one ligated subunit on each dimeric half molecule. Okay, this symmetry rule translates the configurational isomers of heme site ligation. Jay Brown, Kevin Kosek, or one other. Sorry, I'm getting a phone call, a reminder of an appointment. Cooperativity switch points of quaternary transitions. Cooperativity arises from both concerted quaternary switching and sequential modulation of binding within each quaternary form, TNR. Okay, and so by measuring the thermodynamics of the various states, they have come up with a very explicit model for hemoglobin binding to oxygen binding to hemoglobin. Okay, and they use a lot of terminology here: hierarchical code of tertiary quaternary coupling that includes the classical allosteric model as limiting cases. Okay, and so this summarizes in some ways something that I introduced you to in the hemoglobin lecture in medical biochemistry. And we're going to go through some of the data and talk about some of these features. In doing this, I'm trying to expose you to how thermodynamics is used in solving problems in biochemistry, biophysics, molecular biology. Okay. And so here's a rendition of what we're talking about and what I've already alluded to. Okay. These are renditions taken from this paper of deoxy and oxyhemoglobin. Okay, these are basically the Perutz crystal structures in low resolution form. And what you can see is that upon oxygen binding the oxy form, there's a rotation about some axis. The dimer-dimer pairs rotate relative to one another. I think that says 15 degrees. It's a little vague. Okay, and so there's some conformational change that maintains some kind of symmetry, but reflects the difference between these two structures. Okay, and so this is basically the level at which Perutz was working. Okay. And the rotation tends to occur around this GG interface that you can see here in the structure. All right. And they talk about in other places and later, they talk about these various sites and how these various mutations might affect this. Okay. So we have already alluded to, not in great detail, the fact that there were two general models. And the general models are depicted in this paper. Here, I've blown it up here to make it maybe clearer. Maybe it's just as clear here. That there's what is called an all or none concerted model. This is the Monod-Wyman-Janjot model. And there's a sequential model. Okay, this is the Koshland-Nemeth Filmer model. And the idea is that the low affinity form, represented by the boxes, undergoes a all or none transformation to the high affinity form, so represented by squares and circles. And this transition is concerted. All the subunits convert between these two conformations. There's an equilibrium that can be represented here as an equilibrium constant. L is equal to R over T. And then as you add ligands, here represented as S substrates, there's a shift to the high affinity form that isn't represented here, but makes that equilibrium shift more towards the high affinity form. So this is known as quaternary structural changing, okay? And it's represented in a higher resolution than this is this figure before it here, all right? The Koshland model, the sequential model, said, no, no, no. When a ligand binds, that's when a subunit changes to the high affinity form. It only changes in the subunit that binds the ligand. There are some conformational interactions between this R form and the T form of the other subunits, but basically this is the only change, and it occurs sequentially. When the next ligand binds, that subunit changes, and then the next, that one, and then finally the final state. So the changes here are predominantly tertiary, not quaternary. That doesn't mean that there aren't some quaternary changes or interactions between other subunits, but basically this is a contrast between a quaternary all or none model and a sequential tertiary model, okay? And so this is the contrast between these two models. Now, we're mostly going to talk about Gary's ideas that eventually merge these two ideas into some kind of a hybrid model. All right. Now, one of the techniques that they used in work and primarily done in the 70s was to look at a phenomena that mostly I never tell you about, and you probably don't know about except for when you've heard me mention it, that the hemoglobin tetramer actually dissociates, and it will dissociate between a tetramer here, T, and a dimer, D. Okay, and so these data sets cover the concentration range, and I mentioned this, I guess, last week, from 40 nanomolar to 0.1 millimolar, okay? And I didn't do what I think I said I was going to do last week. We need to look up the concentration in red blood cells. In red blood cells, hemoglobin is always tetramer because it's highly concentrated, and therefore the equilibrium that we're looking at here is shifted. The point is that tetramer, oxygen binding of tetramer is cooperative. You can see here that it obeys a sigmoidal curve. Dimers, on the other hand, are non-cooperative. They bind oxygen, but they bind oxygen at two sites, they're dimers, in a non-cooperative fashion. So they're equivalent, they bind with the same affinity, but it's non-cooperative. So it has this sort of myoglobin hyperbolic shape. The affinity is higher in dimers than it is in tetramers. So the lowering of the affinity in the tetramer is one of the ways that it achieves this sort of cooperative transition. So the thing that you are able to show when you study these types of binding curves is that what you're analyzing here is not just oxygen binding to tetramers or oxygen binding to dimers. What you're doing here is you're also studying the equilibrium between dimers and tetramers. Both are going on simultaneously. In the parlance of thermodynamics, the association and the ligation, the oxygen binding, are coupled to one another. You can associate the dimers, or, excuse me, you can associate the dimers, I moved the cursor in the wrong way, two dimers to make a tetramer, or you can bind a ligand, oxygen. The binding of oxygen followed by the dimerization is one path. The association followed by the oxygenation is another path. Okay, and so this is a thermodynamic cycle. It's a path going from two dimers and two oxygens to a tetramer and two oxygens. Okay, and by the definition of a state function, okay, this path must be the same as the association followed by ligation. So the ligation association and the association ligation must have the same free energy change. Okay, for that to be true, therefore, adding up all of the free energy around this cycle must give you the same number. So if you add up twice minus 8.3, because it's binding two ligands, okay, they're both equivalent, so they just summarize it as 2, plus minus 8.8, that sum is the same as 14.3, 5.4, and 5.7. The sum of those numbers is the same as the sum of these numbers. That's the path independence. Okay? Yes, Cindy, you raised your hand. Do you have a question?
No.
No? Okay.
Thank you.
Okay. Over here on the right is this cooperative free energy term. And what that suggests is the difference in free energy going down one path for oxygen binding and another path for oxygen binding, okay? And so 2.9 plus minus 8.3 will give you 5.4. 2.6 plus 8.3 minus 8.3 will give you 5.7. So the cooperative energy is the cost, okay, of the oxygen binding that is reflected in these sums, okay? It is the term that is related to the constraint that I was talking about, keeping it in a certain form, T form in this case, keeping it in a certain form to bind oxygen. And this is the shift that occurs going from the high affinity dimer to the lower affinity tetramer. That's the difference in free energy between these two pathways. Okay, you'll notice the first, second, third, and fourth become more and more favorable as you go down the cycle to the fourth ligand, right? And so you can see that in the binding free energy. Minus 5.4 for the first, minus 5.7, minus 6.7, minus 9.1. So as you go down, the fourth oxygen binding is the most favorable. This is the so-called oxygen cascade that we talk about in that other lecture. Okay, and so you can see that the sum of all of these energies add up, and that this number is the sum of this number minus 6.3 is the sum of this column here. Okay, and so this is the cooperative energy that makes these two pathways equivalent. Okay, and so it's the cycle that gives you this information. Now, here's the important thing to recognize. All right, you can study oxygen binding to get this information. You can study association to get this information. Okay, so if you study association in the presence of different ligation states, then for free, because these are cycles, you get information about the ligation state or the free energy of oxygen binding. So by studying association, you get information about oxygen binding. By studying oxygen binding, you get information about association. Okay, they necessarily inform one another. So if you are doing experiments, you can gain the same information by doing dimer tetramer association, or what I think they refer to as assembly energetics, and infer what the oxygen binding affinities are. And this was sort of the trick or the secret to a lot of the studies that they did subsequently. Okay, and so recall that and think about that as we're going through this. Okay, now I have repeated this, but all I've done is I've stolen some text. The major manifestations of cooperativity found in the heme site are as follows. Dissociated alpha beta dimers exhibit high affinity and bind with no detectable cooperativity. That's the hyperbolic curve. Assembly of dimers into tetramers leads to a large reduction in overall oxygen affinity, as indicated by the right shift. So going from dimers to tetramers, it shifts to the right, so the affinity has decreased. It's a higher oxygen concentration. The tetramer binds ligands with successively increasing affinity over the 4 steps. Increasing affinity, -5,4, 5,7, 6,7, 9,1, okay? And so this adds up to what they refer to as a free energy balance. That's another way of talking about this. I'm talking about it in terms of thermodynamic cycles. Okay, the binding free energy in this lower paragraph, the binding free energy at each step is thus a sum of intrinsic affinities of the sites between ligated and the cooperative free energy term for that step. Okay, the energy cost, and the energy cost is this cooperativity free energy out here on the right. Okay, and so this cooperative free energy, the cost of the protein structural rearrangement that gives rise to the cooperativity. All right, now it looks like you've lost something here, but what you've gained is that ability to switch from T to R state. When I say that, I mean high affinity, low affinity, not tetramer, that you wouldn't have if you just had high affinity. You wouldn't be able to go from 100 millimeters of mercury to 20 millimeters of mercury. I'm imposing what the units would be if we were doing this in terms of millimeters of mercury. So 100 out here and 20 down here that we talk about going from lungs to tissue, that wouldn't occur without this setting up a cost due to structural rearrangement that then allows you to have this oxygen cascade. Okay, and so they're basically describing this into the implications of what's going on. Okay, now we're going to think about this in a much more specific way, and what they represent here are the isomers, all of the different ways that hemoglobin can bind oxygen. Okay, and this is sort of the simplest way that you can reflect this. For our purposes, for their purposes, we're no longer necessarily making a distinction between alpha and beta. So the first ligation state is X's on alpha. The equivalent would be X's on beta, but we're not treating this as separate species. And they're giving it a numbering, okay? And so the numbering is 0 for none bound, and then 1, 1 and 1, 2 for one bound in 2 different orientations. And then 2 ligands bound in a 1, 2, 3, 4 orientation. So you can see it's 2 on the left, 2 across on alpha beta, across alpha alpha, and across beta beta. These represent the 4 simplest ways of representing ligations there. And then a 3, 1, 3, 2 and a 4, 1. So this gives you the simplest way of representing what they refer to as 10 ligation species. This is the simplest way of thinking about this. All right, so this is an important point. What are the minimum number of species that you have to identify? And can you find some difference between them that has implications for the intermediates? Remember that slide at the very beginning. What about the intermediates? So this is deoxy. This is oxy. Are these 8 other guys important or not? Does it matter? Yes or no? Are they different in some way? Are these T form or R form? Is this T form? This is R form. And what are these? What are the differences in these? And that's partially what they're setting out to do. They're setting out to explain how you go down this pathway. Okay? How you go down this pathway going from 1 ligand to 2 to 3 and then finally to 4. When does a quaternary structural change occur? Does it occur when the first oxygen binds, when the second oxygen binds? When? Okay. So the contrast with Perutz was, Perutz basically looked at the structure and then he tried to predict a path. He said, look at the structural rearrangement that's going on here. I predict, and I'm making this up a little bit, but he basically said, I predict that what's going on here, and I'm not seeing my cursor, here we go, is it does this, and then it does this, and then it does this, and then it does this. That's what I'm predicting. And he basically tried to designate a path going from deoxy to oxy. Okay. The problem with this is that it is not statistical. It is not equivalent in terms of possibilities. Why is this one the first one? Well, because Perutz said it's more consistent with the structural change. Because energetically, this and this are equivalent. They should occur with equivalent possibilities. And then going from any one of these to the next one, they also should be equivalent. Remember, we derived that N choose K parameter. And now where am I going to write that down here? The N choose K that involved N factorial over N minus K factorial over K factorial that had to do with the number of ways of putting 2 ligands in 4 sites, etc. Okay. These are basically saying that all of these isomers that must exist are equivalent. They all have the same intrinsic K. They all have the same intrinsic K, and you can't really separate them from one another. Okay, that's basically what this is arguing. Okay, we'll come back to this in a few moments, but it's basically saying, no, 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 there is no unique path. There is every potential path going through isomer to isomer to isomer that is potentially allowable. 1 ligand isomers to 2 ligand isomers to 3 to 4, all of those must be in play and all of those must participate. And that's basically what this is trying to represent. Okay, so on some level, this is an assertion. This is a hypothesis, okay, that this is the logical thing that must happen. This is sort of a reflection of what the Monod-Weinman-Changeau derivation is telling you and what we went through. Now, let me point out one other thing, and then I'll explain something in the lower left, okay? When oxygen binds here to alpha 1, because we designated dimer 1 and dimer 2, alpha 1, beta 1, alpha 2, beta 2. That's basically their arrangement in the tetramer. When the oxygen binds to this site, this species is in equilibrium with none bound, and it's in equilibrium with 1 bound on another site. In other words, the lifetime of this species is not long. You can't isolate 1,1 easily. It's just not stable. There's no way to isolate oxygen bound to hemoglobin in different configurations from one another. At least that is the thought, and that is the truth when you're just dealing with oxygen and iron. All right. What the Ackers lab set out to do was to find tightly bound ligands or metal substituted hemes that mimic oxy and unligated species in a way that allows you to isolate them. And if they could isolate them, then they could study the properties of just this guy, okay, or just this guy, or just this guy, okay? So if you had ligands that didn't fall off, then you could figure out ways of maybe isolating them and studying them and ask, does this guy make dimers to tetramers the same way as this guy, the same way as this guy? Are the oxygen rules the same for all of these species? Is there secondary, tertiary, quaternary structure, T or R, and are there differences between all of those structures? That was the goal that they set out, okay? Unachievable with just oxygen and iron. And then once they set out to do that, they then had to have methods to study those states. One of the methods they used was something called cryogenic quenching. And this allows you to freeze the species in some state and then isolate it from other species in a way that allowed you to learn something about their thermodynamics. I'll show you examples of this and explain that a little more. Okay, so they were basically doing kinetic studies on mixtures of intermediates, watching those intermediates equilibrate, and then learning something about their association properties. And then they also did some chemical cross-linking to stabilize some of these species. Okay, so this is a depiction of what I'm talking about. All right, so basically, notice they're using the term here at the beginning of the legend, thermodynamic linkage between assembly and ligation, okay? There's a linkage between the ability to be ligated and the assembly into tetramers, all right? And I've already pointed this out to you. The path going through ligation and assembly is the same as the path going through assembly and ligation. The sum of the thermodynamic terms must be the same. And if you add up these delta G's, you get the same answers, okay? And so that was the idea. If I can study assembly of dimers into tetramers under conditions where I have different amounts of ligation, then I can dissect out all of the energetics in the system, okay? And so the idea was, let's mix stuff that we can isolate. So we can isolate hemoglobin with no oxygen. We can isolate hemoglobin with 4 oxygens under conditions where they don't dissociate. So this is this high metal, high affinity metal or heme modified structures. There's a table down below. If I mix these two, over time, they will dissociate occasionally into a dimer. At high enough concentrations, this occurs very seldomly, but it occurs. And then the dimers with ligand and the dimers with no ligand will reassociate and make a mixture. So this is a thermodynamic system. And if you wait long enough, and literally the off rates for this are very slow, these equilibrations would last for days. They would incubate them for 48, 72 hours and longer under some conditions, okay? And then quench it. They would usually quench it in ethylene glycol at minus 25 degrees, and then run it out on an isoelectric focusing gel and isolate the 3 bands. And there would be 3 bands because these mixtures, let me see over here, parent species. These mixtures were made up of slightly different tetramers, and the tetramers had slightly different sequences and slightly different isoelectric points. And so these 3 guys would run differently. and you would see them as 3 bands, but the intensity of the bands would tell you which one was more stable. Is BB more stable in AA, more stable in AB, or what? And remember, the assembly thermodynamics gives you information about the ligation thermodynamics. And so it would give you that information essentially for free because of the thermodynamic cycle. Okay? And so between this equilibration, high affinity ligand binding sites, they're metal substituted, and then long incubations followed by cold quenching and then running it out in this weird electrophoresis apparatus at minus 25 degrees. You have to do weird things to the gel and the solution so that things don't freeze and crack. And then you would end up with this equilibration, the fraction AB over the fraction AA and fraction BB. I won't go through all the thermodynamics of this or all of the mathematics of this, but basically it would give you information about their relative stability and give you some information about the relative stability of those different ligation states. Okay, I'm going to skip this. This is basically a figure that concluded that the numbers that they were coming up with agreed with some data, okay? And I swear there's a math error in this, that I should not be in the denominator. All right, so here are the systems that they used. So it turned out that they were able to find that there's a combination of iron and cyanide, and iron and manganese, and iron, manganese, and cobalt and iron. And under these conditions, they could create long-lived oxygen species that they could isolate in methods like this that I just outlined here, study their relative assembly, and then look to see what they see. Okay, and so you can see it in what's depicted here. Not all of the data was collected, obviously, but what you can see to their surprise was there aren't 2 states, there are 3 states. The unligated species had a free energy for dimer tetramer formation of minus 14.4 at 7.4 under these conditions. But then the singly ligated state and one of the doubly ligated states had free energies of assembly of minus 11. And then all of the other species had free energies around 8.3, 8.4. And the same pattern was true at a slightly different pH. And the same pattern was true in the manganese and the manganese cobalt mixture. 15 to 13 to something around 8, 14 to 11, something around 8, okay, 10 to 8, something around 7. And so all of these different conditions gave the same sort of pattern. There is not just a T and an R state. There's an intermediate state. There's a third state that they can see in the system. Okay, so this is depicted in a slightly different way in the other paper. It's the assembly free energies. For the 0, 0 state, it's around 14. And then these 3 ligation states, it's around 11.5. And then all of these other ligation states, it's around 8.5. And if you plot the cooperative free energy that's implied from this, remember that was that other column that was the difference between the assembly free energy and the ligation free energy. It also obeyed the same rules. If we define no ligation oxygen free as 0, then the free energy of cooperativity for this first class is 3 kilocalories cost. Remember, it was a cost for the conformational change, and then 6 kilocalories cost for all of these other states. All right. And so this was a window into something new. This was basically saying, ha ha, when oxygen binds, it goes through this intermediate state. Remember, we were looking for intermediates, where with one ligand bound, and in one case, two ligands bound, there's a free energy of cooperativity cost that we don't see in the other ligation states. And again, notice what we have here, 10 ligation states that were depicted earlier. And this is another way of representing these data. Okay, so they went on to do other stuff, okay, at the same time. Remember I said, now that we have these various species depicted here in this code, no ligands, one ligands, the two ligands, the three ligands, and the four ligands bound, what the total free energy cost is, or the free energy of cooperativity, and notice it's always a cost, it's positive, falls into three classes. 0 for the unligated entirely, around 3 kcals for these three, and around 6 kcals for the other 6. What happens when we measure the Hill coefficient? The Hill coefficient being that midpoint in the Hill plot. Well, the Hill coefficient for this species has a value of around 3.3 or around 2 or around 1. There's a transition that occurs between these different ligation states, okay? The midpoint, P med, shifts in terms of millimeters of mercury. Reactivity of some cysteine groups shift. So there's a local conformational change that makes this cysteine more reactive. And notice it's done in terms of kinetics and the half time of reactivity. Reaction time is long, reaction time is fairly long, reaction time is short. So there's local conformational changes that are occurring. And then we haven't talked about CD yet. We're going to be doing that pretty soon, but CD, circular dichroism, is essentially a measure of tertiary structure. And you can see that the first 4 states are in the T state, and the last 6 states are what they call an R state. And so there must be some characteristic aromatic CD structure that corresponds to T and R. The aromatic CD refers to tryptophan, tyrosine, phenylalanine conformations. And so there's some local conformational changes that must occur. And then the Bohr protons, how many are released? Okay, and notice it's a negative 0.5.7 versus a positive 0.9. So the Bohr proton reflects loss or gain in protons, or gain or loss of protons. I think that makes more sense in the R state. And so what we have is consistent with lots of other biochemical data. The first 4 ligation states are in what is called a T pattern in terms of its secondary structure and reactivity and assembly free energy. And the bottom 6 are what we refer to as R patterns, okay? And so this allowed them to come up with different kinds of ways of thinking about oxygen binding to hemoglobin. There's a reference in the abstract and here to cooperative switching. And all they're really basically saying is, yes, there are pathways as you go from unligated to ligated, but there seem to be specific favorable pathways energetically, not made up by the way Perutz did it, but energetically, there are pathways that allow you to designate, yes, this is a pathway that seems to be preferred or favored, going from one position to another position. I think I'm going to come back to this after we talk about something else, or maybe not even come back to it. Okay. And so this is the insight, therefore, that they gleaned. Okay. Reproduced, blown up, so it's maybe clearer. There's a code for cooperativity in hemoglobin. The first oxygen combined at either an alpha or a beta site, and it stays in the T state. The second oxygen can bind, but as long as it binds in the same dimer, so this dimer on the left, it stays in the T state. And remember, there were 4 different molecules in the 3 classes. There was one that had no ligands, and these are the 3 that had 1 or 2 ligands. As soon as you cross the interface between the dimers, in either way here in the 3 other classes, then it switches to R. Then suddenly you're in the R state. And that's basically what this is saying. These 4 structures, these 4 ligation states, only have oxygen or nothing in one dimer. Once you cross so that both dimers are involved, you perturb the interface in a way that triggers a T to R concerted transition. Okay. So this sort of introduces the way that this was summarized in Stryer and the way that I introduced this in medical biochemistry. Monod, Wyman, Changeau basically say oxygen can bind, but there's a shift in the equilibrium from the T state to the R state all along here. And it can occur anywhere with some gradual shift in the equilibrium. The sequential model says, no, no, no. It's local, it's tertiary, some minimal quaternary interactions as you go down the path. Okay. What the Ackers model says is, this doesn't occur along all of the pathways. The first ligand stays in the T state, it's tertiary. The second ligand stays in the T state if it's in the same dimer. Once it crosses the dimer interface, then it shifts. And so this is what Stryer called a hybrid model. I'm not sure that the Ackers people use the term hybrid model, actually. Okay. But this is basically correct, that it distinguishes tertiary and quaternary conformational transitions, that there are tertiary changes in these first couple of steps. Once it crosses the interface, there are tertiary and quaternary changes subsequently. So initially it's sequential, and then it becomes concerted. Okay, and so that's what this model depicts. All right, by measuring the thermodynamics, by measuring the energetics, you can see that there's a quaternary change only when ligands are across both dimer interfaces. Okay, and you put numbers on it, details on it, in a way that you can see it in terms of stability of the dimer tetramer equilibria, stability in terms of number of protons that are lost or bound, in terms of the Hill coefficient, in terms of the Bohr effect, in terms of local conformational changes in the aromatic groups, in terms of subhydral reactivity, et cetera, et cetera. Okay, so they've tied everything together. All of the conformational stuff, all of the quaternary stuff, all of the biochemistry of specific residues in the structure. Now, as you read this and the other paper, I just grabbed a bunch of terms and a bunch of phrasings: conformational and inner subunit contacts, tertiary, quaternary. Okay, so conformational is meant to refer to tertiary, inner subunit contacts or assembly is meant to refer to quaternary. Cooperativity or self-regulation. Okay, so that's a terminology that you can assign to this and you can think about. Yes, it's allosteric. Yes, it's cooperative. You could also use the term self-regulation. Transducer of free energy. When I introduce, and I think I said this to you before, when I introduce thermodynamics in medical biochemistry, I say Why do we even waste our time studying this? And part of the answer is to understand why metabolism works, to understand how energy is transduced or transferred or converted from one form to another. In hemoglobin, the conversion involves conformational and quaternary. It involves tertiary conformational changes and quaternary structural changes, all of which are linked, thermodynamically linked to oxygen binding, to the energy of ligation, okay? Binding energies are paid for by subunit interactions and conformational interactions. So the ligation, the oxygen binding, is linked to dimer tetramer equilibria and tertiary quaternary conformational changes within those structures. If the free energy in the binding site, the free energy in the binding site can be transduced into a conformational change, the free energy in a conformational change can be transduced into a binding site. They're reciprocal. Another way of looking at this is you can actually take derivatives and look at changes in assembly, changes in oxygen binding, and they can be reflected in things like Bohr effects and the change in the Bohr proton with conformation, or the change in the Bohr protons with ligand binding. They're all dependent on one another, and you can't separate them from one another. Okay, so the Koechlin rule, it only changes conformation when ligand binds. Okay, it's not entirely true, but it's partially true for the first 3 or 4 states. Symmetry rules, that's the term that was originally sort of implied in the Monod-Wyman-Changeau paper. Okay, and that's what these people are now re-emphasizing. The Ackers group is re-emphasizing. Tertiary constraints. Once you are in the T state, different subunits tend to stay in the T state, and the way that they stay in the T state is through this energetic cost, 3 kilocalories or 6 kilocalories. I want to come back. Let's do it. Let's do it here. There's a nomenclature that shows up that is not highly emphasized. Sometimes in these papers, the Monod-Wyman-Changeau paper and the Ackers papers, they sometimes use small t's and small r's to represent different conformations locally, okay? And so by the criteria that I just defined, this is actually not a T state, this is an R state. This is a T state. This is an R state and an R state. This is a T state and a T state. It's the tertiary conformation that's been adapted, and it refers to a tertiary conformation, not a quaternary conformation. So the overall structure is R, but the local structure might still be considered T, although in this particular case, because it's switched, all of these are now in the R state. All of these are basically in the T state, but the local conformation, tertiary, is now in the R state. Okay, and so you have to be careful about making sure you distinguish some of those. All right, and you'll notice that they actually have done that here. Okay, this is actually T because it's still a square. Here they're all R because they're all circles. Okay, so they're now distinguishing between quaternary structures and tertiary structures in this depiction. All right, something that the original Monod-Wyman and Changeau model did not do. did not allow for. Okay. The switch
Dr.
point, yes.
Dr. Correa, could you go back to slide 20? Is that from the T state to R state is less favorable? Because when I look at on the right side from 0, 3 kcal and 6, so when hemoglobin bind like more oxygen, is get less favorable for it to bind to
No, but you're
oxygen?
looking at this figure back here that measures the energetics. Okay. And the oxygen binding is this step. So that last step is 9.1. That's the oxygen binding. Okay. This cooperativity refers to the difference between binding to tetramers and binding to dimers. Okay.
Okay.
And so it's really just a sum of the free energies accounting for the fact that they're linked to one another. Even though you're studying tetramers, it's still in equilibrium with dimers. And there is still a measurable equilibrium constant between tetramers and dimers. And the ligation, the oxygen binding, is slightly different. And that's where the cooperativity comes from. Okay, so in the tetramer state, the oxygen binds tighter than in the dimer state when you're going from 3 to 4. When you're going from 2 to 3, the oxygen binds tighter in the dimer, excuse me, than in the tetramer. And that's the cost, 1.6. That's the difference in the cooperativity. So it's really just keeping track of the thermodynamic cycle, keeping track of the free energy around the box. Does that make sense?
So I have one more question. Is that from Tx2 to Tx3? Is that a endothermic? No, exothermic.
This is telling you the free energy, not the enthalpy.
Okay.
Okay.
Thank you.
Okay. So down here at the bottom, these switch points between quaternary transitions, it's basically saying when you go from, and I think I can do it now. Now, where is it? Where do I want to do it? When you go from here down to here, those would be transitions. So going from here to across the dimer, or going from here to across the dimer, that would give you a T to R. So these are the switch points. Going from this form to that form, that would give you a switch point. Okay, and so that's where these switch points come from. All right, and it's the idea that whatever causes where a ligation switches from T to R, that's would be the transition that causes a quaternary structural change. Okay, so concerted means all or none, sequential means ligand induced. Again, if you remember back to the medical biochemistry lectures, the sequential referred to hemoglobin. It implies ligand induced, but recall in enzymes, we also use this ligand induced conformational change for substrate binding to an enzyme active site. And so there was a ligand induced activation of the enzyme. That concept also came from Koechlin in a different paper. So the take-home message here is that there's a coupling between assembly and oxygen binding, what we refer to, the word we use is ligation. There's a thermodynamic linkage between assembly means dimer tetramer, and ligation, okay? And they're done in a way so that if you measure one, you get information about the other, necessarily because they're thermodynamic cycles. So if you study oxygen binding to dimers, you actually learn something about oxygen binding to tetramers, or you learn something about assembly. Okay. So this, I think, comes out of Van Holt. All right, and so this figure that I moved up above was in that other paper, and it's a different way of depicting the 3 energy levels, and it's the way that they introduce these rules across the dimer interface. And you can see it in the assembly free energy, you can see it in the cooperative free energy, okay? They're inseparable from one another, or they're linked to one another, okay? This is that same depiction where the assembly and the ligations are coupled to one another. And if you go around the pathway one way or on the other way, if you add up all of the free energies, you should get the same answer. All right. And so here in this paper, they give some rules that I think is just a restatement of the things that I've said, but I'm going to still read them. There are 10 species, each with a structurally unique combination of ligated and unligated subunits, distributed into 3 discrete free energy levels. Okay, and it's all relative to the 0-1 species. Okay, procedurally, the cooperative free energies are calculated from experimentally determined free energies, and the sentence is cut off. Conclusion 2, the doubly ligated tetramers distribute into 2 separate levels, which rules out the 2-state MWC mechanism. This feature precludes all purely concerted end-state mechanisms, since they require that cooperative free energy depends solely on the number of ligands bound and not on the specific configuration of the ligated state. By contrast, the actual distribution indicates a dramatic combinatorial aspect to the apparent switching code. Going from T to R is what we're referring to. So it depends on both the number and the configuration. All transitions to the 3rd cooperative free energy level identified with the R are observed in following a general rule. Switching into the 3rd level occurs whenever a minimum of 1 bound ligand exists on each side of the interface between alpha-1 beta-1 and alpha-2 beta-2. This alpha-1 beta-2 interface contains the inter-subunit context in the unligated T-state tetramer and an additional loose contact. And so in some depictions, there's a discussion of where the interactions are. In our previous lectures and in Lippincotts, there's some general depiction between ionic and hydrophobic interactions across the interface, okay? The states that they focused on in some of these studies were the 0-1, 2-1, and 4-1. And so notice it's nothing. This is still a T state because it's within the dimer, and then this is switched to R. And then if you make mixtures of these, then you can get different combinations by studying the reassociation of dimers into mixed tetramers. And then you can isolate these by various methods and learn something about their stability and energetics. Okay. And they did this for those 3 species as a function of pH. And what they discovered was that the assembly free energy of the T states followed this smooth transition in T state where the pH dependence brought it down in a sort of a linear fashion. That the 2-1 state, which is still T, but slightly different energetics, also followed this smooth transition. And then the R state had this really weird break in the curve where it followed a different pH dependence. Okay. This was all done in the so-called cyanomet. This was the first column on the left up above, if you look at the table of all of the different high affinity forms. And so this was another way of studying the difference between the 3 energy levels for the hemoglobin tetramer. Okay, the 2 T states that are separated by 3 kcals, or the R state, one reflection of it as the fully ligated state. They follow a different pH dependent stability. Okay, And then they looked a little bit further at this. And now, Cindy, they give us something about the delta H and delta S, energetics of quaternary assembly. And this has to do with monomer dimer. Notice the proton coupling. So this is the Hill coefficient. Excuse me. This is the Bohr proton. You can see that the T states have a negative 0.6, 0.5. The R state is positive 0.9. Then the enthalpies are negative 30, 33, as opposed to negative 11. And the entropy change is around negative 60. They're slightly different, whereas the R pattern is significantly different and smaller. Okay. And then they do one more thing and they look at mu. Oh, no, here's a different, I'm forgetting this one. That's where I thought I was going. They're showing a comparison for these different species of the assembly free energies and the cooperative free energies. They follow the same sorts of patterns. And then they do a series of mutants. And they do a series of mutants in the structure and how those mutants affect assembly free energy. So you can mutate various residues in various positions, and it affects the ability to make an equilibrium between dimers and tetramers, but it follows this sort of smooth transition similar to a pH dependence. They've obviously been ordered so that they follow the same pattern. This is the higher species, no oxygen. This is one of those species that has, I think, 2 oxygens, but on the same side. And then when you get to the R state, the mutants don't follow the same pattern. You get energetic contributions all over the place, consistent with the fact that the mutations influence the tertiary quaternary structure in a different way, that there's a different structure. And you can see that in the mutation information as well. Okay. Now I add a reference here without going into all of the details. I can make this available if you want to see it. I think I mentioned this this year. It's in this journal, Perspectives in Medicine 2013. It's basically a summary of 1,000 mutations in hemoglobin that have phenotypes. What that means is that they've been isolated in patients, and those patients have some kind of a noticeable phenotype. Okay, now the classic example of this is sickle cell. All right, and we know why it does that in particular patients. Ethnically, it's primarily African Americans or Africans who have this mutation. Sometimes it's in Middle Eastern individuals, and it affects deoxyhemoglobin assembly into fibers that causes red blood cell conformational changes. But it turns out there's lots of mutants out there that have been discovered. And what this paper looks at is the fact that they can have implications for all kinds of stuff. Okay, and I think the summary here doesn't detail all of this. It does not. But basically, it can affect oxygen binding. There can be high affinity mutants or low affinity mutants. It can affect dimer tetramer equilibrium. So it could affect more dimers in the solution. Therefore, it doesn't bind oxygen as cooperatively. You can imagine how that affects oxygen transport. Or it could affect conformational stability. So the tertiary fold of the monomers is not as stable, and so it doesn't make stable dimer tetramers. And so it affects the ability to transport oxygen by that mechanism. Okay, and so it turns out that there were a whole series of classes of mutations and where they are in the structure, et cetera. So if you played the same game that you've been playing with John and looking at binding sites and looking at metal coordination complexes, this would be an interesting place to go look. So this was a summary basically of all of those properties. Okay, everything else that I have in here, I stole out of Van Holt. And so I mentioned that these are just things that were stolen from Van Holt. So we're going to make a transition here. What time is it? It's 11:20. So I'm stopping the sharing and I'm coming back to you. There's a chat over here. And Cindy asked for a break and didn't speak up. So you can speak to me, Cindy. So let's take a break right now and then we'll come back. You want 5 minutes, Cindy? Or you've already taken the break without telling us?
No, I have not. I promise.
Let's take 5 minutes and I'll come back. I won't change anything from this screen, okay?
I'm back.
Okay, we all back? Cindy is back. Ah, looking at the wrong thing here. Okay, so before I transition to something else, I wanted to ask if there were any questions about all of the stuff that I just talked about. It was basically a summary of primarily the science paper, a little bit the PNAS paper from the Ackers lab. And it was basically a deeper dive into how they study oxygen binding to hemoglobin and what they discovered. It emphasizes or re-emphasizes the importance of the thermodynamics and the concepts that we talk about. When I say that things are coupled, when I say that things are linked, when I say that things are a thermodynamic cycle, these are the implications. That by studying self-association, dimer to tetramer, you can actually learn something about oxygen binding. By studying oxygen binding, you can learn something about dimer tetramer. They're not inseparable from one another. They're an equilibrium thermodynamic system. All of the examples in the Monod-Weimann-Changeau paper, there's hemoglobin and everything else are enzymes. All those examples are equilibrium systems, even though they're enzymes and they catalyze a process. And so a lot of those systems, although in the 60s, the stuff that was characterized, it was not that advanced, a lot of it was actually known and understood. So the symmetry idea, the crystallization idea, particular structures, high affinities, low affinities, homotrophic, heterotrophic interactions, that was all known in a lot of these enzyme systems in those days. It wasn't completely understood, and it wasn't as fine a resolution as what I'm showing you for the hemoglobin, but a lot of this was already known in those days. And so you could think about all those systems with exactly the same nomenclature, exactly the same understanding. So do you have any questions at all about what I presented to you today so far?
I don't at this.
I can't hear you, Lindsay. Are you speaking?
Yes, sir. I just said no questions for me at this point.
Still can't hear you. Where?
Oh,
Huh. Your microphone is supposedly working. Why am I not hearing it? Oh, wait a minute.
I can hear her. Oh. Okay.
I can't hear a thing you're saying.
fortunately, it wasn't important. I was just chiming in that I don't have any specific questions at this point, but I'm sure I'll have some later on after I've had more time to look at the material.
Okay. The reason I do this sometimes is to mute people. Okay, so let me go back to and I think so the last slide was that one out of Van Holt, and I wanted to go back to that, but it was actually one of the slides in last week's presentation, and so that's what I'm going to actually I think I have to share. I'm still taping. I think I have to share and then open. Now see your screen and open. Here we go. So in the Van Hold section, the last figure, or one of the last figures he shows in this section, is from hemocyanin. And hemocyanins are oxygen binding systems in other organisms. In this particular example, it's a West Coast shrimp, because Van Hold was in Corvallis, Oregon, University, or Oregon State, I think, actually. And he studied oxygen binding to shrimp. You can study hemocyanins are in various kinds of shrimps or crabs or lobsters or worms or a number of different organisms. They're not tetramers. They tend to have higher ordered structures. They tend to have structures that involve 7-mers. And then those 7-mers assemble into bigger stuff. So they could have as many as 60 subunits. Okay. They bind oxygen cooperatively. And so Ken and a bunch of people, primarily in Oregon, there was another group, the Bonaventuras, were doing this at a research center in Carolina. And then there were people, actually the people I got my PhD with in Connecticut were doing this in Woods Hole in Connecticut. They were studying limulus hemocyanin, which is horseshoe crabs. Okay, and so there's oxygen binding in a cooperative way. And this is depicting a hill plot, nu over n minus nu. Remember, this issue I keep raising with you is what's n on a log scale for oxygen concentration. And you can see the cooperativity between a T state, low affinity. It crosses the 0, point here at a high concentration, and the R state, high affinity. It crosses that 50-50 point at a lower concentration. Okay. The point that's being made here is that the cooperativity is influenced by other stuff. And that was just emphasized in the hemoglobin slides that I just showed you. It's also influenced by pH. And so this is showing you plots of pH effects. And it's also influenced by things like magnesium concentration. Okay, these are also allosteric effectors. Okay, and this comes from a paper from Ken Van Hold and his collaborator in the late 70s. Okay, and so there's a whole parallel field out there. People don't tend to study hemocyanins as much anymore that revealed similar features. Okay, and these allosteric effectors are not just the pH of the blood matters, not just cofactors like DPG, which have metabolic significance, or not just cofactors like carbon monoxide, which has significance for mammals, but other things like salt concentrations, anything that might allosterically affect the assembly state, the quaternary structural state. And you can imagine all of this now in terms of tertiary structure and quaternary structure. Okay. And so this slide is included, this figure is included in Ken's chapter to make this point, but it's also to make a transition. And I want to discuss that transition now in terms of proton binding. Okay, and so we derive Henderson Hasselbalch in our previous lectures in medical biochemistry. And Ken does it here too, but there's an extension that we want to make. So notice that the binding term here is something called R bar, and it's equal to the numbers of sites minus the actual V bar, which has to do with the same equilibrium binding constants reflected as a binding isotherm, but it's reflected as a dissociation constant, not an association constant. Okay, and so It's reflected in numbers of protons bound, okay, divided by this denominator that we got to because we eliminated the concentration of macromolecule, the P term. And so we get this N in our previous nomenclature, which I'm going to write over here. It was NaK 1 plus AK. Okay, and so here it's reflected as the proton concentration, because that's what we're interested in. We're interested in dissociation constant, so it's 1 over K, okay, where this is equal to 1 over K. All right, and then he simply derives an expression. He defines what R is. He defines what nu is with this equation, number of protons bound. And then he plugs this value in here, creates a common denominator, and then derives a new expression, okay, for R bar. All right, and the new expression basically is in terms of a dissociation constant over the concentration of protons. So things flip around a little bit. It's another way of expressing the binding. You can express binding as association or dissociation. And then when he does that, notice we now have a term for R bar, and then we want a term for N minus R bar. So we have both of those potentially here, okay? And he then defines this in terms of a fraction associated and a fraction dissociated. And this was a transition that we made when we did the Hill coefficient, showing that you can convert this to alpha over 1 minus alpha by dividing numerator and denominator by 1 over n. And when you do R bar over n and n over n minus R bar over n, this converts it to alpha over 1 minus alpha, okay? And when you do the math, you'll get this expression. And then this follows perfectly along with what we derived previously for a Hill plot. And so the Hill plot here is log of alpha over 1 minus alpha, log of the left term is equal to the log of the quotient, and the log of a quotient is the difference in the logs. So it's the log of K minus the log of the hydrogen ion concentration. Okay, and so we can reflect this as the definition. The log of the hydrogen ion concentration, or minus the log of the hydrogen ion concentration, is the definition of pH. And the log of K is the equivalent of minus the pK, because the pK is minus the log of K. And so it's minus a minus here to get the plus K. And that's shown down here. All right. So this actually is Henderson-Hasselbalch. If you write it as pH is equal to pK or pKa, bring the pK to the other side, plus the log of alpha over 1 minus alpha. So this is essentially a different way of writing down the Henderson-Hasselbalch equation. And what it tells you is that Henderson-Hasselbalch, in fact, is a different form of the Hill equation. That's actually what it is, reflected in terms of dissociation, not association constants, okay? And so that's the expression that we derive here. All right, just showing you that proton dissociation and binding of ligands to a site can be reflected in the same way. Now, when we think about proton binding to a protein, we have different classes of protonatable groups. We've talked about those before: aspartic acid, glutamic acid, lysine, arginine, histidine, cysteine. there are multiple groups in a protein structure, and they all obey their own Henderson-Hasselbalch equation. And if we reflected it as R bar, we could write down an equation defined up above for R bar in terms of the different groups. This might be arginine and lysine, and then there'd be another one for histidine and cysteine, et cetera, et cetera. And so the R versus pH reflects the loss in proton binding. At low pH, everybody's protonated. As we change the pH, we begin to lose protons. And you can fit these data to basically the sum of the number of protons lost with given pKs. And this is done assuming that all of the groups, all of the lysines have the same pK, all of the arginines have the same pK, et cetera, et cetera. But notice that the plots are slightly different when we change the salt concentration. This is the ionic strength. And as you change the salt concentration, you get shifts in the curves. This is depicting the net isoionic point or isoelectric point is actually way up here, but a point where there's zero charge on the protein. So what's being plotted here is lost protons, but it's also reflecting the net charge on the total proton, or the total protein, okay? So there's a salt dependence to this binding. And so the way to think about the salt dependence is to reflect the free energy in terms of two terms: an intrinsic contribution, which we will call the pK, and an electric contribution, which will reflect something having to do with the ionic strength and the environment in the solution, okay? So it is basically the amount of extra work done against some potential field to remove that proton. How much work does it take to take a proton off of its group and move it some distance away so that that group doesn't see the proton any longer, okay? So if we write the delta G as minus RT ln K, then we can rearrange this to put it into a form where it's log K minus log K, which is PK. In terms of an intrinsic PK in the second term, we're dividing through by RT now, so they canceled. And then this electric term, it reflects 2.303 RT. Okay, so notice we've changed the log and a 2.303 came through in all of these terms. So this is the term that corresponds to the salt dependence. The salt essentially screens the group so that the proton doesn't see the functional group, the aspartic acid carboxyl, for example, as well. And so the charge-charge interactions become changed by this. This is reflected in terms of this definition, where that term, notice the 2.303 isn't included there, in terms of minus W times Z, the average net charge on the protein. So the idea is that the average charge on the protein is seen by the proton when it approaches the carboxyl. And this is the electric contribution. The W is a positive parameter that depends on the dielectric constant and the ionic strength. So it's the salt concentration. It's the fact that it's water, if it is water, okay? And so can the proton see the charge? Does the entire charge of the protein influence that group? That's the assumption that's going on in this. And so you get this additional term. This is 1 over 2.303 times WZ. So this is substituting for the electric term into the equation up above. So what this is saying is that the Henderson-Hasselbalch equation that we use, pH is PK plus the log of the fraction free and bound, okay? That expression has an additional term. And that additional term is reflected in the salt dependence. Its additional term is reflected in the salt dependence, okay? And so let me just depict this here in the corner. Imagine you have a protein. There's my rendition of a protein. And you have a carboxyl group here that is trying to see and bind a proton. And the affinity of this proton for this carboxyl group is influenced by the charge on the protein. Now, it's reflected as an average charge. It's quite obvious. And so this is coming from the depiction of charge-charge interactions that we talked about early on when we went through that chapter in Creighton, chapter 4, where it's sort of point charges that aren't actually true. It isn't really a charge at the center. It's a charge depicted over the entire surface. And some of the surface might be positive, and some of the surface might be negative. And the charges around this particular group might also influence this. But it's pointing out the fact that this PK, or this effective PK, is influenced by the charge on the protein, specifically the charge close to the group, and terms that have to do with the dielectric constant and the ionic strength. If we use mu for the ionic strength, the salt concentration. So as you change the salt concentration here, this proton binding will be affected. Okay. You can imagine that if there was another carboxyl group close by with a negative charge, that can also influence the attraction of this proton. So there's a specific sort of attraction that's going on here. By protonating this group, you minimize this repulsive interaction. So that makes this more protonatable. If you had another group here close by that was a histidine that was protonated, then that would diminish the PK or diminish the attraction here explicitly. All right. Because this positive charge wants this group to stay deprotonated. And so there's an energetic contribution that is far more specific, distance dependent, but far more specific. So the surface of a protein has this complicated stuff going on. What's the total average charge? What are the local charges? What's going on in the water? Is it really a dielectric constant of 80? What's the salt concentration? And how is that influencing this particular feature? All right. And so when we do titrations like this, all of that basically has to be taken into account. We treat it as though all the carboxyls are the same. They're not. All the histidines are the same. They're not, because of the reasons that I'm describing here. It's much more complicated and much more specific. And in fact, the problem that I gave you from Van Hold, where he changes the charge on the DNA by 80% because the salt concentration is diminishing the charge by 80%. You assumed 100 millimolar, and then you recalculated the Donan effect term for the virial coefficient. That's exactly what I'm talking about. Salt binding to DNA, sodium particularly, changes the charge on the DNA, makes it less charged, and therefore there are fewer charge-charge interactions between DNA, double-stranded DNA, and other DNA molecules and other substances in solution. And this small section at the end of this particular chapter is one way of thinking about that. It becomes clear that proteins frequently fine-tune the PKs of important residues, as in catalytic sites, by the placement of other charge groups in their vicinity, and by the binding or the interaction of other ligands. I can't get down to that corner on my screen. So if you have anion binding, for example, to those groups, this will also fine-tune the interactions that are going on. And we know, for example, that anion binding in particular is one of the things that regulates electrostatic potentials in proteins and protein-protein interactions. So let me close that. Let me stop sharing here. Okay. So this little aside hopefully ties together something about that particular problem set that you were working on for I guess that's problem 6. Yes. All right. Now, do you have any more questions? Once again. Okay. So we're now about to take a week break. Okay. There's another assignment out there. It's assignment 7. I made a version of it where I copy pasted the figures out of the book. I'm not sure that that helps or hurts. It turns out that chapter 14 and the chapter 15 version have different numbering. So I gave you the questions out of chapter 15, which was the PDF copy of that book. There's 6 questions related to binding and solving binding problems. All right. We're scheduled, according to the syllabus, to start talking about spectroscopy the next time. I think I'm going to also use a chapter out of Creighton. It's chapter 5. If you go to Canvas, at the top section, there are some references there. The copy of the Van Holt book is there. Chapter 4 from Creighton is there, and chapter 5 is there. So it's just basically an introduction to protein structure. It happens to also include something on CD. So I'll use it as a way to integrate a refresher, if you will, on protein structure. It's maybe more detailed than I've talked about. I don't know if you've covered this in any great detail in the cell biology class. I think you haven't. But we'll use it as a way of sort of integrating what we want to talk about. And then the absorbance fluorescence CD is a chapter out of Van Holt, and that's what we'll end up talking about in 2 weeks. Anything more? Okay. I'm waiting for some homework. I'm waiting for some reattempts at corrections to 5 and 6, and now I've given you 7, which won't be due until the 22nd or something. All right.
Could you
Go ahead.
explain? I mean, I still got stuck on 2.10 in homework number 5.
I know you're all being stuck, but the thing is that none of you actually ever turned anything in that was an attempt. Most of you just sort of moved the equation around in a funny way and never really tried to derive it. I'm recognizing that you're all having trouble, but I sort of don't want to jump in and help yet. I've given you some hints. Let me give you a different hint. The affinity for the KNI is tight, 10 to the 9th. The affinity for the AC is much weaker. It's 10 to the 6th. Okay. So the displacement experiment binds to a site that kicks out the AC. All right. And if that's all that was really going on, then the apparent affinity would actually be something like 10 to the 4th. It would be basically the ratio of those 2 equilibrium constants, because one involves one going on and then one going off. And so the overall equilibrium constant would be a quotient. And it would apparently be 10 to the 4th, 10 to the 3rd, maybe, is that weak. But that's not what you see. What you see are weaker affinities, but not 10 to the 10th all the way down to 10 to the 3rd. You see 10 to the 10th to 10 to the 9th, the 8th, the 7th. The reason for that is that AC does not bind to all of the HIV proteins. It only binds to a fraction of them. Its affinity is 10 to the 6th. It's micromolar. And you're doing experiments in the micromolar. You're not doing experiments in the millimolar. You're not saturating all of the sites. So some of the KNI is actually also binding to free sites.
Okay.
Okay. And so the equilibrium involves a mixture of free site binding and competition or displacement. And so there's something else happening. And as you titrate with AC, it modifies what's happening because more and more of the sites are occupied. And so it diminishes the overall equilibrium for the K and I. So that's what's really going on in that equation. Okay. That's a hint. All right.
Okay.
That's a
Thank you.
hint. I admit that all of these, like the paper I sent you from Maurice, I don't know if some of you know who Maurice is. I think he still is a dean up there. They just pluck it out of thin air. Here's the equation. Okay, so see if that helps. Think about that. All right, and then when we come back, we'll attempt to fill in the gaps here. The other thing I'm going to do is I just got this morning. Somebody sent me a series of videos. The videos are from somebody named, I think her name is Nancy Pye. Yeah, her name is Nancy Pye. She's apparently an MIT math major, math graduate maybe, who makes these short videos on calculus. They're short, 10, 15 minutes, and it's just practicing how to take derivatives under different circumstances. Somebody else sent it to me because he's been playing with this stuff. So I'm going to send you a link to Nancy Pye videos. He sent it to me by text, and I'm having to figure out what the right link is for Nancy Pye. Some of you don't need this. Some of you need this. Okay, you need more practice. And so I'm going to share that with you. I think the one that I tried to share with you was a longer video by a different guy on deriving derivatives and deriving the formulas. This is literally just, here's the rule, learn the rule, apply the rule. Okay, which may be more useful at this point than figuring out where the various rules in calculus come from. Okay, so I'll do that sometime soon. I was trying to do it earlier, but the links were these really long things, and I didn't want to send you that. Okay, so I'm going to stop, and I'll see you in 2 weeks. And let me see if I can